Good afternoon, distinguished guests. As the last speaker of this meeting, we'd like to invite Dr. George Cotsarelis. He is an outstanding researcher and clinician in hair biology and hair disorders. Today, he will talk about stem cell in androgenetic alopecia. George, please.
Thank you. Thank you for the organizers for inviting me, and I really want to thank you all for staying till the bitter end. Appreciate it. I hope I'll make it somewhat worthwhile. I'm going to talk about our research on androgenetic alopecia, and specifically male pattern baldness. So this is really, to me, a totally fascinating topic. Don't let anyone tell you that it's not. If you think about what has to happen for the hair to go from this to this, it's pretty remarkable. And the main process, of course, is miniaturization. We have a large antigen. We start with a large terminal antigen hair follicle, which only through cycling miniaturizes with each successive progeny being smaller, follicle being smaller and smaller. And histologically, of course, it looks like this. You can see the bulb way down in the fat, whereas in a miniaturized follicle, the bulb can be all the way up here where the sebaceous gland is. The other important characteristic morphologically is that the sebaceous gland enlarges in androgenetic alopecia. So this, I think, really demonstrates where most of the loss of the volume is occurring. This is a horizontal section of the terminal follicle, and you can see the outer root sheath here, inner root sheath here, and the hair shaft. And this is a miniaturized hair follicle, and you can see the outer root sheath, inner root sheath, and then a tiny hair shaft. So most of the loss seems to be in the hair shaft. I've been asked for decades whether stem cells are involved in androgenetic alopecia, and early on I knew that through studies where we grafted human skin onto skid mice and did label-retaining cell studies looking for quiescent cell populations, we would see these heavily labeled cells that had taken up label 4 months previously and retained it, indicating that they hadn't proliferated, were very quiescent. So there was indications early on in the mid-90s that there were very quiescent cells in the bulge area of the miniaturized follicles. And then when we found a marker, K15, for the bulge cells, staining with that showed a pretty prominent population within the follicle. Here's that little tiny hair shaft. But we really wanted to quantitate the number of stem cells in male pattern baldness, and Luis Garza was someone who came to the lab who really wanted to do this in a very careful way. And so essentially what he did was he compared scalp from the occiput of men undergoing hair transplants to the recipient areas on the same subjects. And he isolated the epidermal and hair follicle epithelium and stained with antibodies to stem cell markers and the facts. And first he wanted to show that K15 was a good stem cell marker in these human cells. And we knew from work done in Howard Green's lab many years ago that the size of a keratinocyte correlates with its proliferative potential. So small keratinocytes with a high nuclear cytoplasmic ratio are the ones that can be passaged many times and are so-called holoclones. So to look at whether K15 expression correlates with cell size, we did this kind of analysis where looking at the basal keratinocytes using alpha-6 integrin antibodies, you can see that the cells that are at the highest 5th percentile for keratin expression they're also the smallest cells, so a good correlation there. And then we wanted to look at whether K15 expression correlates with quiescence, because that's another characteristic of these hair follicle stem cells. And again, in the highest 5%, you can see that these are cells that have the highest percentage of G0, or the resting phase of the cell cycle. So we felt confident that by looking at the top 5% of K15 expressing cells, we're looking at quiescent and highly quiescent cells with a high proliferative potential. And so when we did our analysis, here's an example, we were pretty surprised to find that the stem cell population in the same individuals, looking at balding scalp and haired scalp. And I should mention that this picture is really just to remind us that we're talking about androgenetic alopecia. The patients were not, completely bald, Hamilton 6, 7. They were really the ones undergoing hair transplant. So there was a lot of transitional areas in there. But the stem cell population was retained. And so we went to look more in depth at markers, and fortunately Manabu Oyama, when he was in Jonathan Vogel's lab at the NCI, did elegant laser capture on human hair follicles and found CD200 as a marker for the bulge cells, and this was confirmed by Inoue. And when we looked at CD200, we found a population of cells that was markedly diminished in the balding scalp. So these cells had very high levels of CD200 and alpha-6, and you can see that they were quite depleted. So what are these cells and where are they? If you look at their K15 expression, about 85% of the cells had expressed K15 at substantial levels, suggesting that they were in the bulge. About 15% expressed this marker BREB4, which is a nice marker of the secondary germ, which is right below the bulge. So, in other words, they were scattered between the bulge and the secondary germ. What about their stem cell characteristics? Well, the cells that were diminished in the balding scalp were not as were a little bit larger, not as small as a true stem cell population, and then they were also more proliferative. The true stem cell population was almost all in G0. These were more proliferative, indicating that they may be descendants of these more quiescent cells. So this data really suggested that there was essentially a cell type that had characteristics of a progenitor cell that may have arisen from a stem cell that was more proliferative and gave rise to progeny that would normally populate a large follicle. And we also looked at another marker, CD34, which in humans, as Bruno Bernard showed a couple days ago, also that the lower outer root sheath below the bulge stains for CD34. And if you look at CD34 cells in balding scalp, they're also markedly depleted. If you look at their stem cell characteristics, these cells have very low levels of K15, almost none, and also they are very large. So they're quite different than the true stem cell population. So this suggested to us that stem cells are intact, progenitor cells are decreased. And so possibly there's a defect either in stem cell activation, either because of a lack of an activator or the presence of an inhibitor. And at the same time, we were interested in looking directly at bald and non-bald scalp for gene expression with affymetrix arrays. And again, work done by started by Yaping, and Luis jumped in and analyzed the data more closely. And this is the kind of thing you've seen over and over, but basically each row here is a gene, and you can see, of course, that there are differences between the haired and the bald scalp. One gene that came up as very highly differentially expressed was prostaglandin D synthase, which was increased 7-fold in the balding scalp. Here are 4 more patients, not the same ones used for the arrays, showing that bald scalp had high levels of PGDS. And then the protein was also elevated in balding scalp compared to haired scalp. So what is PGDS? So it's an enzyme that converts prostaglandin G2 and H2 into D2. And so you would expect that D2 would also be elevated, the lipid, the prostaglandin. And in fact, that is the case using ELISA in different patients. Again, you can see there's a marked increase in the prostaglandin D2 levels in the balding scalp. So just to give you a little bit more background about L-pgds and prostaglandin D2, we know that GDS is a testosterone responsive gene, and that it actually is responsible for virilization in the brain. Minoxidil, there's some data that might be working through prostaglandins. And prostaglandins, of course, everyone knows, are known to influence hair growth, and there are products commercially available for eyelashes that are essentially either analogs or in this case, the actual prostaglandin F2 alpha. So the other thing to keep in mind is that in other systems, prostaglandins have sort of a yin and yang effect. So there are often prostaglandins that have opposing functions. So for example, in the lung, bronchial muscle tone PGE2 causes dilation of the bronchial muscle tone, and D2 causes constriction. So our hypothesis really was that we knew that E2 and F2 are sort of pro-hair growth, D2 might be inhibiting this function. So one of the first things we did was to actually look at the ratios of all the different, or the levels of all the different prostaglandins. So you would predict that if DS, this enzyme, is increased, you would have an elevated level of D2, which I showed you, but you would also predict that E2 would be decreased because the reaction is going this way, and that J2 would be increased because this is a product of D2 that's non-enzymatically produced. And that is in fact the case when we collaborated with Garrett Fitzgerald, who's at Penn, who's a prostaglandin expert, and looking at D2, J2, these are both increased in bald scalp, and E2 is actually decreased compared to haired scalp. So, but one thing to keep in mind is that D2 is the predominant prostaglandin in the scalp. The absolute level of D2 is higher than the other ones. So at this point, I've shown you that PGDS, mRNA and protein are elevated in the bald scalp. D2, the lipid, is elevated in the bald scalp. And E2, sort of the good prostaglandin, is decreased. So the next question was, do prostaglandins have a functional role in hair growth? And so we decided, since the mouse is such a nice model of hair follicle cycling, we just wanted to look at expression of the enzyme L-pgds as well as the lipid prostaglandin D2 during the cycle. And when of course, you're all familiar with the idea that the mouse cycles synchronously. The hair follicles go into anagen and stay there during the first 17 days of life. And if you look at levels of D2 and DS, the levels of DS actually increase slowly until the end of anagen when they go up. And then, as you would predict, after DS goes up, D2 levels go up subsequent to that. This is the spontaneous hair cycle. If you look at the depilation model that Ralph House popularized many years ago, this gives you a sort of a crisper peaks here because the follicles are even better synchronized. So at the end of anagen, you get an increase in DS, and then during catagen, you have high levels of D2. So this suggests that prostaglandin D2 is playing a role during the regression stage of the hair cycle. And if you look at expression of PGDS, it's really in the outer root sheath, starting right below the level of the bulge, and in that sort of CD34 positive area in the human follicle. And so it's in an area that's not in a stationary area. In human follicle, it's actually in the same area. And I should have mentioned that this is late antigen. So if you look at day 12 or 13, you're not going to see this kind of staining. It's only at the end of antigen. And in the human, when you see antigen follicles that are labeled, they look like this. Very similar pattern below the bulge. The interesting point here is, one interesting point is that when histologically you look at a scalp biopsy, you look at antigen hair follicles, you don't know whether those antigen follicles have been in antigen for a month or a year or 2 years. And so this is probably a marker of late antigen in human hair follicles. So similar to FGF5, which goes up at the end of antigen in the mouse, this enzyme seems to be expressed in a subset of human hair follicles that I assume are probably towards the end of the antigen. And if you look at other follicles that have smaller follicles, for example, you can see that, again, it's not only is it in the outer root sheath, but interestingly, it's also in mast cells. So just keep that in mind. We'll get back to that in a minute. So in order to look functionally to see whether these prostaglandins actually inhibit hair growth, Luis did some experiments where he depilated mice and then 8 days later began to treat topically with prostaglandin D2 and J2 or vehicle. And then he measured the hair length at day 12. And so what he found was that the mice that had received the D2 or the J2 had shorter hairs compared to the vehicle. So this was inhibiting the growth of the hair. And D2 is known to bind to 2 different receptors. They're called PDGDR and GPR44, otherwise known as DP1 and DP2. And we were fortunate to be next to Garrett Fitzgerald, who had knockout mice of these receptors. And so we thought that it would be really valuable to determine which receptor the D2 is working through in order to inhibit hair growth. And so the experiment here was the same as the one I just showed you, but on the knockout mice, Luis applied the D2 topically. And what he found is that the mouse that lacked the GPR44 or the DP2 receptor, there was no difference in the hair length when D2 was applied, indicating that there was no inhibitory effect. So whereas the one with the DP1 receptor knockout, there wasn't an effect. So it looked like the DP2 receptor was the one that was important for this inhibitory effect. So we're always interested in, of course, the human and the human hair follicle, and this was work done earlier by Gurpreet, who's currently at Allergan, but he was at Gillette when he did this work, and basically we collaborated with him and he had shown that D2 in the Philpott assay, the explant type of assay in human hair follicles that were microdissected out and put in culture, that when you added D2 to the culture media, there was an inhibition of hair growth shown here, and J2 even had a more profound effect. Both of these were statistically significant. So this indicated that D2 was inhibiting human hair follicle growth, both from men and women, and in this assay. And so we went on to use an inhibitor of the D2 receptor in culture, and basically we're able to show that we could reverse the inhibitory effect of D2 when adding this inhibitor. So again, keep that in mind for a second. This pathway, looks pretty complicated, but you can see that it all depends on arachidonic acid being converted to G2 and H2. And earlier, Susan Fisher, interested in carcinogenesis, had developed a mouse where she overexpressed COX2 or PGS2 under the K14 promoter. So this mouse was making a lot of COX2 in the epidermis and the follicular epithelium. She was interested in carcinogenesis, but what she noticed was that the mice had hair loss. And so we got these mice from her, and this is what they looked like. When you look histologically, it was really remarkable because here's your wild type mouse with a nice long follicles going into the fat. This COX2 overexpressing mouse had miniaturized follicles and really large sebaceous glands. So pretty striking phenocopy of androgenetic alopecia. And when we analyzed all of the lipids in this mouse skin, D2, PGD2 was the predominant prostaglandin that was present in this mouse. J2 was also elevated. E2 also elevated, as you would predict based on this, where this is so far upstream that you're getting increases in both sides of this pathway. So basically, we think that prostaglandin D2 contributes to androgenetic alopecia. I've shown you that there are elevated levels of D2 in human scalp that's undergoing balding. And I've shown you that this mouse that has very high levels of all these lipids also has hair loss and has miniaturization of the follicle as well as sebaceous hyperplasia. So L-pgds expression also suggests that this enzyme and lipid is important for the regression stage of the hair cycle. And in a human explant model, D2 clearly inhibits hair growth, and using the inhibitor to the DP2 receptor, we can rescue that inhibition. The thing that's really exciting is that there are at least 7 drug companies that have developed inhibitors of DP2, the receptor for PGD2, and they were doing it because, as I mentioned before, these were found on mast cells, and so they were trying to develop drugs to be used for asthma and for allergic rhinitis. And so a few of them actually went into phase 2. Most of the studies were anywhere from 4 to 6 weeks long, and they really had no effect on asthma or allergic rhinitis. So they were essentially abandoned. But they're out there, and we think that, well, hopefully our goal is to really try to resurrect these, and given some of this preliminary data, try to develop them into a new type of treatment. I've been very fortunate to have really excellent postdocs and trainees in the lab. This work, as I said, was primarily driven by Luis Garza, who's at Johns Hopkins right now, assistant professor there, and Yao Ping did all the array work early on, and she's currently at Merck. So I just want to, again, thank everybody for staying till the last minute, and thank my funding source, and I'll stop there. Thank you.
Thank you, Dr. Cotsarelis. He mainly talked about prostaglandins in hair biology. Any question, comment, please?
Yes, thank you for this talk. I have short 2 questions. One is, did you see any hair phenotype in the prostaglandin D receptor mutant mice without adding prostaglandin D2? And the second question is, did you try to check hair elongation in the field post model only with the receptor inhibitor without the prostaglandin D2?
Yeah, so the first question was, did we see a hair defect in the receptor knockout mouse? And the answer is no. And then the second question was, in culture conditions, we did not see an effect with just adding the inhibitor.
Any other question? George, in your experiment, first progenitor cells, the population of progenitor cells decreased, but in your theory, eventually at the end of boarding, the stem cell in AGA also decreased or disappeared.
You're asking whether the stem cell population
Yes.
decreased when
Eventually.
Eventually, yeah. So
Yes.
we never used skin from someone who's completely, bald. Again, looking at staining and a lot of sections, it's pretty remarkable how many stem cells are present, even in the tiniest miniaturized follicle.
Any other question about the prostaglandins? Complex dual effect? Okay, thank you very much, Dr. Cotsarelis.